Greeting friends. So far we have discussed about what is gluconeogenesis. In this video we are going to discuss the important reactions of gluconeogenesis. Okay, before we discuss these reactions, let's discuss an important point. Suppose this is A and this is B. We want to go from A to B, but that's not possible due to some reasons. I will tell you that. We develop a way from where it is going to A to B. Why we can't go from here to here? Because there is a hurdle and we cannot cross this hurdle because you can say this is too high. Okay, we can't jump that. So we develop this pathway, this strategy from where we can go from here to here. Same is the case with these gluconeogenesis reactions. Okay, let's see how many reactions in glycolysis. 10 reactions in glycolysis and 10 reactions in this pathway also, gluconeogenesis. So what is the difference? Although there are 10 reactions in each of these pathways, but they are different. 7 of these reactions of glycolytic pathway are same in this gluconeogenesis, but 3 reactions catalyzed by the, we can say, pyruvate kinase and the PFK, and the third one is hexokinase, are totally different. So these 3 enzymes which catalyze the irreversible steps, you can say, in glycolytic pathway are different in case of gluconeogenesis. Why they are different? Because of this reason. What we are doing here is we are bypassing. So that important term is bypass. We are bypassing this hurdle. Okay, so the same thing we are going to do in these reactions, because we are going to bypass these reactions through some other way. Why we are bypassing? Because of the irreversibility, we can say. These reactions are irreversible, and because of that these reactions are highly exergonic, so we cannot do that. Okay, so let's do what is here. Okay, so the first thing is, here is the reaction. The first reaction is conversion of this pyruvate to PEP. This is the first bypass step, and this is consist of 2 reactions further. So this pyruvate to phosphoenolpyruvate conversion consists of 2 reactions which are occurring in different compartments. So different compartments, that is the 2 reactions. One of these 2 reactions would be in mitochondria side and other would be in cytosol. Okay, and then what more is the 2 routes. So there are 2 routes. What does that mean? It means we can make this phosphoenolpyruvate through 2 precursors. Okay, that is pyruvate here. This is the first thing, first route, pyruvate or alanine. You can use both, and this would be converted into PEP. And second is lactate. Lactate, remember, lactate is produced from pyruvate when there is shortage of oxygen. That is referred to as, we can say, anaerobic glycolysis. If you haven't watched this, you can go and check that video also. So what we are going to do here is pyruvate or alanine. Why pyruvate or alanine? Why not only pyruvate? Because alanine is this is our alanine. Okay, this is a amino acid. What we are going to do here is we are using transaminase. That transaminase would remove this NH2 in the form of this, and we would end up with pyruvate. So this is our pyruvate. So we have found pyruvate from alanine also. Okay, so we can use this alanine or pyruvate for the formation of phosphoenol pyruvate. And the next is this lactate, which is produced in anaerobic glycolysis, that can be also used to make PEP. So these two routes are different. Okay, so the next point here is the two routes is done. Before we actually discuss the details of these reactions, let's make a layout for these reactions. That is what we are going to study in these reactions. Pyruvate is there. Okay, this is present in cytosol. Remember, I told that these reactions are occurring in different compartments. The first reaction that is in the mitochondria and the cytosol. So this is in cytosol first. This is sent into mitochondria now. So pyruvate is in mitochondria now. In mitochondria, it is converted into oxaloacetate. So this is converted into oxaloacetate in mitochondria. So before it is sent again into cytosol, it is converted into malate. Now, why there is a need to convert this oxaloacetate into malate? There are two reasons for that. First, I will tell you now. The second, I will tell you later. Okay. The first reason is because there are no transporters on the mitochondrial membranes for the transport of oxaloacetate into cytosol. So that is why we are converting this into the malate. What we are doing here actually is reducing this. Okay, we are reducing this oxaloacetate into malate. So now this malate can be directly sent into cytosol, where this malate is converted again into what? Oxaloacetate. This oxaloacetate is now converted into PEP, our desired product. This is what we would discuss. Okay, so we are starting now. The first reaction is this pyruvate to PEP, which is consisting of two reactions. Cytosol is there, which was present in, we can say, initially in cytosol. Now it is in mitochondria. So this is our pyruvate. Yes, this is our pyruvate. What we are going to do? We are going to do is that we have here bicarbonate ion. This is bicarbonate ion. This is pyruvate. We are going to add this bicarbonate ion here to make it oxaloacetate, and the enzyme here is, this is our pyruvate, and this is, you can say, a carboxylic group, carbon dioxide molecule we are going to add. So this is pyruvate carboxylase, the enzyme which is going to add carbon dioxide molecule, you can say simply. Okay, so this is there, and in the process of this reaction 1 ATP is used, PI. And very important point here is that this reaction requires biotin. This is a direct question for your exams. This for the enzyme, that is this pyruvate carboxylate, but does this enzyme require as a cofactor, and that is biotin. And biotin is a carrier for this HCO3 negative. This biotin is carrying this molecule. So what is here? This is, we can say, like this now. This is our oxaloacetate. I must draw in blue color here. Okay, so this is the first reaction in mitochondria. Now this oxaloacetate is converted into malate. This is our malate now, and in the process this is oxidized form, okay, and we are going to reduce it with the help of reducing agent NADH, and this would be converted into NAD positive. So this is reducing this oxaloacetate into malate. So we have produced malate, and this is going back also, and so you have to emphasize on the numbering of these NADH and NAD. So one NADH is used, okay, and this would be transported back, okay, into the cytosol, where there would be a compensation for this NADH positive which was used, and we would produce one more NADH because this is also going back. So the next reaction is in the cytosol, where this oxaloacetate would be converted into our desired product, that is pyruvate, through the action of the enzyme. What is that enzyme? This PEP, carboxykinase. Now this enzyme is kinase. So what this enzyme is going to do? This is going to do the terminal transfer of what? Phosphoryl group from a crunchy molecule, and this crunchy molecule here is GTP, and this is GDP now, and also there would be removal of CO2 group from here. So this is, we can say, let's draw something more. How it is being converted, this oxaloacetate is converted into, we can say, PEP. This is our GTP. Let's draw the structure of the GTP. This is guanosine, okay, attached to 3 phosphoryl groups. So it is GTP, okay. This is going here, you can say this, okay, and this would go here. Next, yes. Now this is a carbonyl group, this particular group, okay. So this is polarized now, and this polarized carbonyl carbon, this oxygen particularly, would attack on this terminal phosphoryl group and would remove it, okay, and the result would be, you can see here, and this is the removal of carbon dioxide. This oxaloacetate would be converted into CH2 double bond. What is there? C and hydride bond of phosphate. Is it okay? Yes, this is okay. This is our COO negative. What is this? Enol form. Phospho is there, and this is pyruvate. So this is PEP. This is phosphoenol pyruvate, isn't it? So this is done now. This reaction is done. What you have to note here is the NADH to NAD positive ratio. This is the particular point we have to emphasize now. Look at this. This NADH to NAD positive ratio in cytosol is 8 into 10 raised to power minus 4, and this is 10 raised to power 5 lower than mitochondria. So we can simply say that in cytosol, we can say the concentration of NADH is low as compared to mitochondria. So the concentration of NADH is low in cytosol as compared to mitochondria. So for that, what we are going to do? We produced this. We used first NADH to convert oxaloacetate to malate, which was necessary because there were no transporters. Once we used that, we converted again. Why did we convert? Because we needed this NADH. If it was not there, then the biosynthesis, because remember, this is biosynthesis of glucose. This is not catalytic pathway now. This is biosynthesis, okay. So we need this reductive agent for the biosynthesis of glucose, because it is already used in converting what is that, 1,3-bisphosphoglycerate. We used 1 NADH in converting 1,3-bisphosphoglycerate into glyceraldehyde 3-phosphate. So we need this NADH to continue the synthesis of this glucose, not glycolysis. Okay, so we can say that the use of NADH and production of NADH is important. See here, because this is the path from pyruvate to PEP. This is important because it keeps balance between NADH produced and consumed in cytosol during gluconeogenesis. Okay, so this is about the cytosolic concentration of NADH, we can say. The next point is here, the formation of pyruvate from lactate. Okay, so this is our lactate which we produced in anaerobic glycolysis. This is converted into pyruvate. Everything is same. We are going to convert this lactate into pyruvate. That is the only difference. And then for this pyruvate is further, we can say, is converted into this PEP, and the reactions would be same. Those two reactions catalyzed by first the pyruvate carboxylase and then PEP carboxygenase. But there would be one more difference, I will tell you. This lactate is converted. This is, we can say, oxidized by oxidizing agent, this NAD positive, and this would be converted into NADH plus H positive. We have produced this NADH in the cytosol. This is occurring in cytosol, and now this pyruvate is going into mitochondria, and the reactions are same as we have seen earlier, those two reactions. But the difference here is that those two reactions are occurring in the same compartment, that is mitochondria. We converted this pyruvate into oxaloacetate, then we converted that oxaloacetate into malate. Okay, and then we transported that malate into, we can say, cytosol. We are not converting this we can say pyruvate which is converted to oxaloacetate, low acetate in mitochondria. Here in this case lactate to pyruvate conversion, we are not converting this oxaloacetate into malate. Why so? Because there is no need of this NADH now, which we have already produced. So why we are going to do the same steps again? Okay, so there is no need to convert this oxaloacetate into malate. So we are going to convert this oxaloacetate directly into PEP, and the enzyme is PEP carboxygenase. So both of these reactions are occurring in the same compartment, that is the mitochondria. So this is the difference. Okay, so I think this video would help.